All right, so welcome to the next in this series of Bitesize Bioinformatics talks, where we're just covering little details of useful stuff that it's useful to know in bioinformatics. Today we are going to take a look at a dynamic JavaScript plotting library called Plotly, but specifically we're going to look at it to use it in context with Ggplot in R, so that we can make dynamic graphs that you can interact with in a really simple way within a normal sort of R session. This is a kind of cut down light version of some of the larger interaction frameworks that exist in R. So the bigger and more comprehensive framework that this is all sort of little parts of is something called Shiny. We're not going to get into Shiny today. We're just going to stick to dynamic graphs with Plotly, but if you're interested in that, we will be doing some more focused training on developing Shiny applications in R in a couple of weeks. So if you're interested in that, just keep an eye out on our mailing list or drop us a mail and we can send you the details of that. So for today, though, what we're going to look at is a simple way of generating interactive graphs in R. And this all comes out as part of a project that's been around for a while now, which is, let me just bring up my screen, which is a project called Plotly. So Plotly started out as a JavaScript library for doing dynamic plots on web pages, and it's still that, but it's now got bindings for other languages, so you can now use the same sort of framework to generate dynamic plots in both R and Python. Okay, today we're going to focus on the R side of this. So if I go through here, you can see the sorts of plots that it can draw. So it has a wide range of plots that it can do. But if you go through the Plotly pages on here and look at the code examples that they have, then what they're going to do is to use their own syntax and their own R packages to draw these plots. But what they also provide, and is actually a much easier way into this type of functionality, is that they provide a function which is a bridge between Ggplot and Plotly. So inside the Plotly documentation, there is this magical function called Ggplotly, which converts a ggplot graph to Plotly. So if you're already familiar with doing sort of more conventional static plotting in R with Ggplot, then using this is a super simple way of transforming those static graphs into dynamic graphs with interaction, which you can then put into your reports. Okay, makes you look super exciting. So we're going to have a look at how we do this. So I'm going to go through this, build up a little script on here so that you can see how it works. When this exercise is over, I will put the contents of the code that I've written in here up on the Bitesize Bioinformatics Google Drive page, so that if you just want to see the output of this or directly reproduce it, I'll put all the data and the scripting up there so you can see it there. Let's start by loading a data set in and just doing some basic plotting in the conventional way that we would with Ggplot and forget about the interaction. We're going to need to load a couple of libraries. Ggplot is part of Tidyverse, so I'm going to load the Tidyverse packages. And then I'm also, even though I'm not using it just yet, going to load the Plotly package as well. So Plotly is the package that provides the Ggplot to Plotly bridge. It's just on CRAN, so you install it with install.packages, the same as you would for anything else in R. Okay, so I'm going to load those two packages in first. Now I'm going to load in, let's not put that there. I'm going to load in a data set, a small one to start with, just so I can see how this is going to work. So if I load in this little data set here, and have a look at it. This is my little example data set that I'm going to work with to start with. So it's just a set of data about the brain weights and the body weights for a whole bunch of different species that have been categorized as either domesticated, wild, or extinct. So we're just going to look at that relationship. Now, I can do this to start with just using a conventional Ggplot. Okay, so if I put in a block on here, I can say that I want to do a Ggplot graph. I set up my aesthetics. This is going to be just an XY scatter plot. So on my x-axis, I'm going to have the log brain weight. On my y, I'm going to have the log body weight. And I'm going to color the points based on the category. Okay, so I'm going to plot the log brain weight against the log body weight and color by category. And I want this to be a scatter plot, so I'm going to use geom_point to draw this. Okay, so if I do that, I will see the standard sort of graph that I get. Let's make the points a little bit bigger. So say size equals 4. Just make these a little larger. We can see the conventional sort of static Ggplot graph that we can draw very easily with R. Now, this graph is great, but there are some limitations in it. So if I draw a graph like this, I might go, okay, well, that's great, but well, what are these 3 points? I know that they're the extinct species because they're colored that way, but I can't tell what actual species they are. Likewise, I might want to know what some of these are. So there's no interaction on this, and there's no way I can click on a point to find out what it is. What we can therefore do is to use Plotly to provide that level of interaction. And it's super simple. In order to convert this into an interactive graph, all I need to do is to firstly save the Ggplot graph into a variable instead of just drawing it straight onto the screen. So I'll call that brain plot. Okay, so I've now created that as a variable, and then just do the ggplotly and pass in the ggplot that I want to convert. And now if I run that, I will get the Plotly version of the graph. So it looks pretty much the same as I had before. It's directly converted, but now if I put my mouse over any of the points, you can see that I've got interaction, and this is actually a dynamic graph. It's not just a fixed picture. We have some basic tools on here at the top, some of which are going to be more useful than others given what we're doing. So one, for example, is that you can say what happens when you hover over the plot. So by default, it just shows you the metrics for the closest point. But you can also have comparative points. So if I just click on here, now instead of just seeing 1 point, it'll show me from a line on the x-axis whichever points are close to that, so I can start comparing between them. Okay, so little things like this make it really useful. Okay, we'll look at some of the other tools that are up there later on. At the moment, this is inserted into an R notebook, but R notebooks, in the end, you can compile down to output documents. And if you can compile this into an HTML output, so if I just go into the menu up here and say knit to HTML, you'll see that not only does this plot stay interactive within RStudio when I'm constructing it, but once it compiles to an HTML file, that interactivity is maintained, so it's still there. And even if I then open this up in the browser, just take the HTML file that comes off there, again, the interaction is still there. So when you're compiling your reports out of the end of this, most commonly you'll compile them down to an HTML file. Now you can just email that to someone else, and not only can they see the data, but they can poke around in it and they can see what else is on there. Okay, so it's a super nice and super simple way to be able to deal with this. Let's look at some of the other things that we might want to do in here. So at the moment, the aesthetics that I've set up in here are just the ones that I need to generate the plot. Okay, I've got the X aesthetic to plot the brain weight, the Y for the body weight, and the color for the category. one of the questions that I sort of said that I would like to answer in the first place is I'd like to be able to pick a point on here and see which species it was. And at the moment, you can see that I can't do that because the points are only displaying the aesthetics that are used for the plot. So it's got the value for the brain weight, the body weight, and the category, but it's not showing the species. So one of the things that I would like to be able to do is to expand the set of information that's presented when I put my mouse over a point in here. And that's very easy to do. All I have to do is to create an aesthetic mapping for an aesthetic that doesn't actually get used by the plot. Plotly doesn't care whether an aesthetic that you've defined is actually consumed by one of your geometries. So geom_point here is consuming X, Y, and color, but I can set up another aesthetic. So I could say species equals species. So this is the name of the column out of my original data frame. And now if I rerun this, even though this isn't going to be picked up by Ggplot at all, so the plot itself looks exactly the same, because it was a defined aesthetic, Plotly will pick it up, and now it will actually list the species in. Okay, so it tells me that this is a Triceratops, that's a Diplodocus, this is a rhesus monkey, that's a chimpanzee, that's a human. Okay, so you can expand the set of information by adding information into these aesthetic mappings, okay, even if you don't use them directly in the plot. We could also go the other way, that we might decide that we've got too much information on here, that if we have a plot that's defined many aesthetics, we don't necessarily want to see all of them. So even though I need the aesthetics for brain weight and body weight to actually draw the plot, I might not want those in my tooltip. If I want to limit what's in there, I have an option within the Ggplotly function itself where I can say tooltip equals, and then I can either pass just a single aesthetic in. So I could say, well, I only care about the species. So I can say the tooltip is equal to the species. Okay, so now that will just show me the species, or I can select multiple ones, so I can pass a little vector of aesthetics in here. So I could say species and category. Okay, so now it's going to show me the category and the species. If you read the Ggplotly documentation, it says that the order that you pass these in is the order that they're shown in the graph, but they're not. So this shows it as category species, and if I switch this round, it does exactly the same thing. So the order here seems to be completely irrelevant. It just does it in descending alphabetical order as far as I can tell. One thing to note in this, though, is that when I've put this in, you can see that I had color as the aesthetic that was marked as category. On here, the name that comes up on here is not the name of the aesthetic, it's the name of the column in the original data, but you can specify either in here. So I've put category up, but I could have said color and species. Okay. Sorry, it doesn't work for that one. Let me change this around. So if I had said, instead of defining an actual aesthetic that I needed, I can make this into anything I like. The Ggplotly will still work on here, and I could say, instead of specifying the column name, the name of the actual aesthetic, and that still picks up as well. So you can either use the aesthetic name or the column name when defining what you want to show, but in the end, it's always the column name that appears in here. Okay, so you can cut this down to whatever you want, specifying either an aesthetic or a column name, but the aesthetic name doesn't actually show up in the tooltip, only the column name does. Let's look at a slightly more complicated example, because that's going to allow us to show a few more bits of information on here. So I'm going to insert another block on here and load another data set. Okay, and if I just have a quick look at what's in here, then this is a slightly larger data set, but it's going to be the same kind of example where I've got some genes under here in a column called probe. I've got two quantitative gene expression values in here, an ESC and an IPS, and then from various stats and stuff that I've done before, I've classified these as either unchanging or up or down. And then I've also got a description for each of the genes. Okay, so let's just go through the same sort of process again, but now with the slightly larger data set. So if I start off just doing the standard Ggplot graph, so I can say RNA, my aesthetics in here, my X is going to be my ESC, my Y is going to be my IPS, and my color is going to be the class that I specified in there. And again, we'll do this as a geom point. Okay, so again, standard static Ggplot plot, and again I can convert it into a plotly function by saving it to a variable and putting it into the ggplotly function. One oddity with ggplotly is that you would think that because all we're doing here is saving this into a variable and then doing ggplotly on the RNA plot, okay, which is how we can draw the plot. You might think that because all we're doing is taking the output of this and putting it as the input of this, that you should just be able to pipe out of the end of the ggplot function and pipe that into ggplotly. But you'll find if you try that, it won't work for reasons that I'm not entirely clear about. It's something to do with the tidy evaluation code, but the bottom line is that you can't pipe out of ggplot and straight into ggplotly. You have to save it to a variable and then put the variable in as the argument to ggplotly. Okay, so again, from doing this, we've got our dynamic plot from that. The ggplotly dynamic plots will pick up pretty much all of the customizations that you would do on your plot. So here I'm just using default coloring on here, but if I wanted to put in a non-standard coloring scheme, I could just modify the plot in the same way that I would for any other ggplot. So on here, if I said scale color manual and then values is red to gray and blue to, something like that, and I run that again, then the dynamic plot will pick up the customizations that I've put in, and I'll get those dynamic colors in here as well. Okay. As before, if I want to change the annotations that come up in the plot, then I can use the same sort of tricks as I showed before. So here I want to put the gene name and the description in. So I can say gene equals, and then the actual column that contains the gene name in here is called probe. And then I can say description equals description to get that in. So that will expand to include those two extra pieces of information. So now I should have all of the aesthetics that I had before, but including the gene name and the description. Okay, so you can see all of these on here. Now, I don't particularly like the fact that I have a gene name in here that's actually called probe, okay, because I want it to be called gene. So if I do want to be able to change the name as it's displayed in here, then I can do that by modifying my data as it goes into ggplot. So because the name in the tooltip is picked up from the column name, if you want to change it, what you need to do is to change your column name before you start into ggplot. So here, if I wanted to change that probe column to be called gene, before I get to ggplot, I can just do a rename on the column to say gene equals probe. pipe that straight into the same Ggplot as I had before, but now my column will be called gene in here. And if I run the plot again, I'll see that the tooltip should now show the actual gene name under a parameter called gene. There you go, instead of probe. Okay, so you can modify it, but you have to modify it by changing the name of the column before you do your Ggplot, and then it will be reflected in here. As before, if I want to limit the parameters that are shown in the tooltip, I can say that only show me the gene and description in there, and that should just show the two values that I want. Okay, so now just gene and description, okay, which can be quite useful. Other things that we can do in here, this is at the moment the only dynamic use we've made of this has been in changing the tooltip on here, but you can also use this to look at subparts of the plot. So if I was interested in points that were down here, they're quite difficult to see because of the scaling of the plot, but I can just drag a box inside here to select part of the plot. And if I do that, it'll actually zoom in on that lower portion of the plot, so I can view that in more detail. And having done that, I can just double click, and that should then zoom back out and show me the entire plot. So the interactivity can be both on zooming and also on mouse over operations. Okay. It's either being very slow or it's, oh, there you go, it does come back eventually. Now, let's look at a couple of other things that we might want to modify in here. We can have a look at changing the size of the plot. Now, this is something that's going to be different from the way that you would conventionally do this in Ggplot, or indeed inside our notebook. In a notebook, what you would normally do is you would go into the header of the segment that you're in, and you would say something like fig height equals, let's make it, actually, let's make it that stupidly big. Let's make it very big. Fig height equals 30, fig.width equals 30. Okay, and if I just did the Ggplot part of this, then you would see that my plot would then get very big. It would cram into the thing, but I would see it all in tiny points crammed down into the bottom. If I do the same thing as that with the Ggplotly, though, then when this draws, you'll see it hasn't changed size at all. Okay, so although you would conventionally change the size of a plot in a document by setting fig height and fig width, that will only affect static Ggplots. It won't affect the Plotly plots. The equivalent for Plotly is that you set the height and the width in the actual Plotly command on here. So here, if I say height equals 800 and width equals 800, the units in here are in pixels. That will then change the size of the plot. Within the notebook document itself, you only have a limited amount of area to draw plots in. So if you make it too big, you'll just get these scroll bars on the end because the size of the plot is bigger than the area that the document has given you to draw it in, which is a bit annoying. But if you compile this into an HTML document, then what you'll see is that at the point where you do the HTML compilation, it will actually keep the size, and you'll get the larger version of the plot in there. So here you have the larger version of the plot in here with no scroll bars and stuff on the side, and all of the interactivity still in it. let's just take that back. So to finish up with, I'm going to look at a couple of other things that we can modify in here. We've said that in general, we are using the Ggplot interface to Plotly because it means that we can just recycle all the knowledge we already have about our Ggplot object. But what we get out of the Ggplotly function is a Plotly object. So we can do further modification of the behavior of the plot by adding additional parameters to the Plotly object. So there are two sort of main things that we might want to change in this, which will be that we can either change some of the configuration of the plot, or we can use it to change some of the layout information on the plot. Now, there's a whole bunch of information for this on the Plotly site. So on here, in the section here, and I'll put this URL into the video when we put it up, there's a whole set of details about some of the behaviors you can put into this, but I'm going to show a couple of options that you can modify by changing the Plotly options in a way that's not possible through Ggplot directly. So if we have a first look, one of the first things that we may or may not want to do is that on the dynamic plots, there's a little menu that comes up at the top that allows us to do things like saving the plot as a PNG, zooming in and out, doing various types of selections and changing the hover modes. If you preferred, you might decide, well, actually in my plot, I just want to keep it as clean as possible and I want to keep that toolbar away from the plot, so I don't want to actually draw that. So we can illustrate that by taking the Plotly object, piping that into a call to config, which is the Plotly configuration function, and then this is a set of key value pairs to say what the preferences are. So in this case, I can say display mode bar equals false, and if I run that, okay, it still draws, but now you'll see that when I put my mouse over here, the bar that used to display at the top now no longer does. Okay, so I can modify that. The other thing that I might want to change as an example might be that at the moment, the tooltips that are coming up are inheriting the coloring from the coloring of the plot itself. So my down points are going red, my unchanging points are going gray, and the blue points are going the up points are going blue. Okay, which is fine, but maybe I don't want that, so I can separately modify the behavior of the additional Plotly pop-ups by changing the layout parameters on this. Okay, so again, I can take the output of that, which is the Plotly object, and now I can adjust the layout. The structure for the layout is a little more complicated. So because in the layout, you have to say what part of the plot you want to configure the layout for. So in this case, I'm going to change the behavior of the hover label. And then you need to say what parameters for that you want to change. And because you might be changing multiple of these, you pass these in as a list. So in this case, I'm going to say that the background color of the list is now going to be white. So what we can see then, having added the extra layout option on here, that now our labels come up in white, and there's a whole variety of things that we could change in a similar way. All of these types of interaction are not just limited to scatter plots, we've shown that on here, but any of the standard plot types will work. So for example, if I just load another small data set and do an example of a bar graph, we can see how that would work as well. So if I do okay, so if I load this small data set up, I could do something similar in here where I can say, let's draw a bar graph from this. So if I say, let's Let's plot the last name against the age. Okay, so that would just give us a normal bar graph like this, but we can save this. And again, although it's a different plot type, same idea, we still get a plot up with interaction on it and where we can add other factors into here. Okay, so that's pretty much all I wanted to cover for this. Unfortunately, because we had a power cut in the middle of this, there's no one left to ask any questions. But if anything comes up from this, this will be up on the YouTube channel in a bit, and we'll be able to pick up from there, and we'll be able to answer any questions if people put them onto the channel there. Okay, I hope you found it useful.